The Public Health Agency of Canada and the Canadian Food Inspection Agency have developed laboratory biosafety guidelines to help public health and microbiological laboratories develop biosafety programs and policies. These guidelines reflect current biosafety and biocontainment principles and practices in Canada. The World Health Organization's guidelines provide biosafety guidance at an international level. There are hundreds of biocontainment laboratories in Canada. Most require fairly low-level containment features and practices. They include university, private, hospital, and government laboratories that handle pathogens which can cause disease in humans and/or animals, but are unlikely to cause serious disease under normal circumstances. By normal circumstances, we assume the person working in the lab is healthy, meaning their immune system isn't weakened for any reason. Some pathogens handled in a Containment Level 2 lab can cause serious illness and even death in immunocompromised persons. As an example, if you have a cold, your immune system is not in its normal state. Keep in mind that if you're pregnant, your immune system is also affected. A lab is designated as Containment Level 2 when its features and practices allow the people working in it to safely handle infectious agents that can be transmitted by ingestion, inoculation, and through mucous membranes. In most cases, the agents handled in a Containment Level 2 lab are not transmitted by the airborne route. The higher the containment level, the more stringent the safety measures. In this presentation, we will cover biosafety management, handling pathogens, working in a biological safety cabinet, and wearing the proper personal protective equipment. We will demonstrate correct techniques for decontamination and handling biohazardous waste. We will also discuss working with cell lines and working with small lab animals. Finally, we will explain various important regulatory considerations concerning Containment Level 2 labs. Just remember, it is everyone's responsibility, yours as well as your lab manager with assistance from the biosafety officer, to use the information and recommendations in these guidelines to do your work safely and securely. Working with pathogens involves a level of risk. Determining just how much risk allows labs to implement operational practices that will help reduce, mitigate, or eliminate the risk to lab workers and to the community. That's why risk assessment is such an important part of biosafety. For all pathogens being handled, each laboratory must conduct its own detailed risk assessment to determine to which risk group they belong. The risk assessment process will also determine the lab's required physical containment level and appropriate operational practices for the work to be done. As a result, the lab's safety and training requirements for the people working in it will also be determined. The containment level is based on the manipulations generally associated with lab-scale and clinical procedures. Processing larger volumes of pathogens, for example, may mean having to work at a higher containment level or with additional operational practices. All containment facilities should have a biosecurity plan based on the outcome of the risk assessment. Formulating and implementing this plan involves a diverse working group, including scientific directors, principal investigators, laboratory workers, biological safety officers, security staff, the responsible official, if one is designated, and law enforcement agencies. Keeping an updated inventory of infectious agents is an important biosecurity measure and a good practice. Every time an agent is added or removed from the lab, that movement should be tracked. Whether an agent enters the lab from another lab, or as a result of a diagnostic test and propagation, or as a result of verification of proficiency panels, it must be noted. The same applies when an agent leaves your lab as a result of a transfer or inactivation and disposal. It's important to know at all times what infectious agents are present in the laboratory, those that are being used in current work, those that are in storage, as well as those that are just sitting at the back of the freezer. Prior to handling pathogens in the lab, a health and medical surveillance program for the lab workers should be in place. The program may include requirements for pre-employment and periodic testing of employees, available prophylaxis, and special considerations. It may also include immunization, such as hepatitis B vaccination, if any work with blood or body fluids is to be undertaken. Everyone working in a Containment Level 2 lab must be trained to follow the Biosafety Manual or standard operating procedures for that facility and for the projects in progress. These SOPs must be compiled in a manual that is available to all staff. SOPs must be reviewed and updated regularly. Training must cover the potential hazards associated with the agents and the work in question, and the necessary precautions to prevent exposure and release of infectious materials. This includes the proper use of biological safety cabinets and personal protective equipment. The training must also include detailed response procedures for emergencies such as spills, accidents, exposures, decontamination, as well as biosecurity incidents. Trainees must be accompanied by staff members when in the lab. Personnel must show evidence that they understood the training. This could include a period of dry runs or supervised work. The training must be documented, signed by both the employee and supervisor, and the records must be kept in a safe place. Regular retraining, including the review of the SOPs, is strongly recommended. The best laid plans, the most comprehensive programs, and the latest equipment are practically useless if you don't use your training or don't follow proper microbiological techniques. Some basic principles apply to every Containment Level 2 laboratory. Food and drinks are not permitted in laboratories. Change your clothes if you are not dressed appropriately for your work. And it's best to remove your contact lenses and wear glasses to work in the lab. Store your street clothes in a different location than laboratory clothing to avoid cross-contamination, and leave personal belongings outside the laboratory. A Containment Level 2 lab must be separated from public areas by a door, which is kept closed at all times. Entry is restricted to authorized personnel only. The signage at the entrance to each lab must specify the safety hazards inside the lab as well as its operational containment level. It must show the entry requirements, such as personal protective equipment or necessary immunizations. It must also provide the contact information of the responsible person. Inside the lab, always wear a lab coat and make sure it is snapped up. This also applies to visitors and trainees. Put on a pair of gloves before setting up and starting your work. This dress code is necessary for your protection in all Containment Level 2 labs. To offer effective protection, appropriate laboratory clothing has to be worn by everybody while in the lab, and it has to be fastened properly. Never wear your lab clothing when leaving the facility. If the clothing's contaminated, decontaminate it before washing it. Sandals or any open-toe and heel shoes are not permitted in any lab. Appropriate footwear will protect you against exposure, slipping, crushing, and punctures. It will also shield you against electrical and chemical injury. When the work to be done may create a risk of splashes or flying objects, do your work inside a biological safety cabinet to contain infectious particles. Otherwise, protect your eyes and mucous membranes by wearing protective glasses and a face mask. In case of an accident, emergency eye wash facilities and emergency shower facilities are mandatory inside or in close proximity to Containment Level 2 labs. You must wear protective gloves for any and all procedures that may involve direct contact with a biohazard. If you're allergic to latex, there are alternatives. Depending on the level of risk associated with your work, when you're working in a BSC, you may consider wearing 2 pairs of gloves. When you don the second pair, make sure you pull the glove over the sleeve of your lab coat so your skin is protected. Always remove your outer gloves before removing your hands from the BSC. When you remove and dispose of the gloves, always make sure you turn them completely inside out. Avoid touching the contaminated surface with your hand or fingers. Change your gloves often and always wash your hands after removing your gloves and before leaving the lab. Make sure you don't carry infectious agents into the environment. In a Containment Level 2 lab, you probably won't need respiratory protection on a regular basis. However, if your lab's risk assessment requires you to use it, you must be trained to use it correctly. The most common type of respiratory protection used in a Containment Level 2 lab is the N95 respirator. Its use is recommended when the risk of exposure to aerosols is high, when working with infected and shedding animals, when working with large volumes or high concentrations, when working with a pathogen that can be transmitted by the airborne route, or when working with an emerging pathogen whose mode of transmission is uncertain. Be safe. The most important thing about respiratory protection of any type is that it has to fit properly, otherwise it is useless. Everyone who wears a respirator at work must undergo a documented training session and should be fit tested annually. Personal protective equipment is just one of many methods for controlling biohazards. To be effective, it has to be chosen correctly, and you have to be trained to use it properly. In general, the equipment provides an effective barrier against infectious agents and can prevent the spread of contamination, but it does have limitations. Biological safety cabinets can be an effective primary containment barrier if used properly in conjunction with good lab techniques. Clean benches and fume hoods must never be used for handling pathogens, since they do not offer the user or the environment effective protection against these agents. There are many classes and types of BSCs that offer varying levels of protection. Once again, the risk assessment will help determine which one is best suited for your Containment Level 2 lab. You should use a BSC that has been designed, manufactured, and tested in accordance with National Sanitation Foundation Standard Number 49. It will bear the NSF seal. It must also be tested and certified at least annually by an experienced, qualified individual, and everyone working in a BSC must understand how it works and be trained to use it correctly. BSCs create and maintain a curtain of air between the environment and the inside of the BSC. Contaminated air is exhausted through high-efficiency particulate air or HEPA filters before being released in the environment. Any disruption to this air curtain caused by movement in front of the cabinet or by sudden movement inside the cabinet reduces its efficiency as a protective device. The first step is setting up the BSC. Turn on the cabinet blower and fluorescent lights. Check the air intake and exhaust grills for obstructions. Confirm that air is flowing inward by holding a tissue at the middle of the edge of the viewing panel. If it's drawn in, the BSC is working. Disinfect the interior surfaces. Remember, you should be wearing gloves even when setting up the BSC. You should place an absorbent pad under the work area to absorb any drops and help reduce the creation of aerosols. Load all materials into the cabinet. You should separate them into clean and dirty work areas. Wait at least 5 minutes before starting your work, allowing the cabinet air to equilibrate. BSCs are really too small for 2 people to work in at once. The work area becomes crowded. More arm movement might disrupt the air curtain and reduce its efficiency. Also, the NSF Standard 49 is tested for 1 worker positioned in the center of the front of the cabinet. This means that if more than 1 person works in the cabinet at one time, it is not known if the cabinet still protects either the personnel or the product. Make sure you don't overload your work area. You can store extra supplies outside the cabinet. The next step is starting your work. Sit and adjust the height of your chair so the bottom of the sash is level with your armpits. Make sure you're wearing gloves. If you need to reach out of the cabinet, take off your gloves first. When adding or removing items, always move your arms slowly and perpendicularly to the front opening. Allow the air in the cabinet to stabilize before resuming your work. Inside the cabinet, limit your movements of dirty material over clean material. Perform procedures that may generate aerosols towards the back of the cabinet. Keep waste and contaminated material inside and toward the rear of the cabinet until your work is done. Never work with open flames inside the cabinet. This can damage the HEPA filters, and the heat buildup can disrupt the airflow patterns. There are alternatives, such as disposable sterile loops and micro-incinerators. Don't use a centrifuge inside a BSC. The infectious particles ejected from the centrifuge travel at speeds too high to be retained by the cabinet's airflow. The airflow patterns of the cabinet may also be disrupted. When you've done your work, close or cover any open containers and allow the BSC to run with no activity for a few minutes. Place all contaminated waste material into biohazard waste bags within the cabinet. Surface disinfect all objects before removing them from the cabinet. Remove your contaminated gloves and dispose of them within the cabinet. Disinfect the outside of the waste bags, disinfect the work surfaces, and regularly disinfect hidden surfaces. Turn off the fluorescent light and cabinet blower. UV radiation doesn't penetrate particles, so don't count on it to decontaminate the interior of the BSC. Nothing can replace old-fashioned disinfection by hand. In fact, we recommend that you do not use nor depend on UV lights at all. Some BSCs must be left on at all times. Remove and dispose of gloves and wash your hands. The Standard Operating Procedures binder must also include procedures for accidents and emergencies. You should know what to do. When a spill occurs inside a BSC, infectious aerosols are generated. Leave the BSC turned on. Follow your facility's standard operating procedure for spill cleanups. Move slowly but deliberately. Remove your gloves and discard them inside the cabinet. Remove your lab coat or gown if you suspect it's been contaminated. Don fresh gloves. Assemble cleanup materials. These materials should be in a spill kit that is ready to go, and put on protective clothing. Carefully cover the spill with absorbent paper and gently pour disinfectant over the area. Begin at the edge of the spill and work your way to the center. Using a gentle flooding action will reduce the creation of aerosols. Use the appropriate disinfectant. In most cases, sodium hypochlorite is recommended for spills. If you suspect the infectious material has spilled onto the catch tray, pour disinfectant through the perforated grill. Allow sufficient contact time, at least 30 minutes, before moving on to the next step. Use forceps to pick up any broken glass or sharps and place them in a leak-proof, puncture-resistant container. Wipe up the spill and dispose of contaminated materials in an autoclave bag within the BSC. If the items can be autoclaved, thoroughly disinfect them before you remove them from the cabinet. Wipe the inside of the cabinet with disinfectant and let the BSC run for at least 10 minutes before resuming your work. Never put your head in the BSC. If the spill occurs outside the BSC, follow your facility's standard operating procedure and apply the same general techniques. If there was a large volume that was spilled, it is recommended that you change your lab coat or gown and exit the lab for an appropriate amount of time to let the BSC filter aerosols. The entire surface of the BSC, including the work surfaces, sides, back, and interior of the glass and catch pan, must be decontaminated. Standard operating procedures must be in place for any other kind of emergency, such as BSC failure, fire, exposure to infectious materials, and escaped animals. Also, you have to record and report all emergencies immediately to the lab supervisor. Keep a written report of everyone entering the facility during the emergency, such as firemen or paramedics. Regarding disinfection, the disinfectant you use must be appropriate to the infectious materials you're handling. And you absolutely must disinfect your work surface, whether it is a workbench or the BSC, prior to beginning your work and at the end of each session, even if you're just stepping out for a quick lunch. Don't assume someone else already did it. All contaminated materials, solid or liquid, must be decontaminated before disposal, reuse, or removal from the laboratory. This is usually done by autoclaving. You must monitor the autoclave's efficacy regularly using biological indicators and mock loads. Remember to use the appropriate biological indicator for the type of material being decontaminated, and place it correctly in the middle of the load to ensure its usefulness. Results of these tests and cycle logs must be kept on file. One last point about autoclaving. To allow steam to penetrate the bag, never seal its opening. Make sure to always use a positive control indicator from the same lot number for validation. Some labs might not have access to an autoclave, and their biohazardous waste has to be picked up by a specialized company. If that's the case in your lab, make sure the waste material is contained to prevent the release of the contaminated content, and make sure it's labeled appropriately. Use appropriate biohazard bins or containers, tape them shut, and surface disinfect them, if necessary, before removal. In a Containment Level 2 lab, chances are you'll be working with cell lines at some point. A risk assessment for each of those cell lines will determine the necessary operational practices. The cell lines you work with might be contaminated with bacteria, fungi, mycoplasma, viruses, or prions even before you've started working with them. The expression of latent viruses is one of the primary hazards of manipulating cell cultures. Most laboratory workers are not aware of these hazards. HeLa cells, for example, contain human papilloma virus. Additional precautions are required when working with primate cell lines because they present specific biological hazards. The same can apply for some recombinant cell lines, which may contain lentiviruses, and cell lines derived from bovine tissues, which may contain prions. You could be working with small lab animals. A small animal is one that can be contained in a caging system: mice, rats, rabbits, and other species. Specific guidance for animals is provided in the Canadian Food Inspection Agency standards and the Canadian Council on Animal Care guidelines. You may have to use extra protection when you enter an animal room. If the animals are carrying or have been infected with a pathogen that is transmissible to humans by the airborne route, you must use respiratory protection. You must be familiar with the shedding characteristics of the pathogen in the animal species. Always wear gloves, but if you know the animals might scratch or bite, then wear special protective gloves. All staff must observe strict lab-specific entry and exit protocols that also cover any exchange between the animal room and the lab. Each animal room must be labeled with its unique hazards and entry requirements, and cages housing infected animals must also be labeled appropriately. Working with small laboratory animals poses unique hazards, bites and scratches, as well as noise, smell, and temperature, not to mention exposure to infectious agents that may be naturally occurring, experimentally produced, or acquired by cross-contamination between the experimental animal and others. When you are cleaning or emptying cages, try to minimize the creation of aerosols and dust. Some facilities have special cage changing stations to reduce this risk. Use the restraint methods you've learned to minimize scratches and bites. Become familiar with the characteristics of the animals you'll be dealing with before you start to work with them. What is their temperament? What are their instincts? What are their physical attributes? But also, which infectious agents could they be carrying? And what is the potential route of transmission? Carcasses and tissues are considered contaminated. They must be transported from the animal room in leak-proof containers that are appropriately labeled, then incinerated or correctly processed. If you work with animals, you need to consult the containment standards for veterinary facilities and the Guide to the Care and Use of Experimental Animals, published by the Canadian Council on Animal Care. The risk assessment process determines your lab's required containment level and operational practices for the work to be conducted. Under certain circumstances, additional operational procedures may apply. These additional requirements are based on the pathogen, its virulence, the amount of infectious material being handled, and on the type of work being done in the Containment Level 2 lab. For example, you may have to wear a full front gown and N95 respirator and gloves at all times. You may also be required to carry out all manipulations in a certified biological safety cabinet as if you were working in a Containment Level 3 lab. This situation is described as Containment Level 2 physical requirements with Containment Level 3 operational practices. There are rules that apply to the transportation, importation, and use of pathogens. First, anyone transporting an infectious substance, whether shipping or receiving it, must be trained in the transportation of dangerous goods as required by Transport Canada. Next, before importing a pathogen into a Containment Level 2 lab, you must ensure that you have a valid importation permit from the Public Health Agency of Canada and/or the Canadian Food Inspection Agency. Without it, the shipment will not be allowed through customs. It may be destroyed or sent back to the shipper. Pathogens that are common to both humans and animals require an import permit from both agencies. For some pathogens, approval must be obtained before transferring these pathogens between labs. These regulations are discussed further in the Laboratory Biosafety Guidelines and in containment standards for veterinary facilities. You can also visit our website for more information. Remember, the most important thing is your safety.